Hello, my name is Wolfgang Wagner. I'm heading the Institute for Stem Cell Biology at Aachen University Medical School. And today I'm giving the lecture within our lecture series Gene and Cell Therapy on Ethical Considerations. Well, in fact, there are many burning ethical questions coming up in stem cell biology and gene and cell therapy that I think need to be considered also by society. And this lecture is conceived to have interaction with the students. Actually, it was a 3-hour course where we had very intensive discussions. Within this recording here, I can of course only tell a bit on how the discussion went and which the focus of the discussion and the impression of the students was. So to start with ethical considerations, I'd like to first point about human embryonic stem cells. The first human embryonic stem cells were described already in 1998, so it's not so super long ago. They were initially generated from spared embryos derived from in vitro fertilization, and they, of course, raised immediately high hopes. These were now cells that were capable of differentiating into virtually all cells of our body, and there were enormous hopes in regenerative medicine that we can use these cells for generating new hearts or livers and organs. Looking back, I think we can say that it's not quite as easy. So there are now the first clinical trials ongoing, but of course there were also many hopes that were just fostered. At the same time, there were also many fears that may have been exaggerated. And I can tell that by the time people were really concerned about how to deal with human embryonic stem cells, because of course they were initially derived from embryos that at this time also needed to be sacrificed to isolate the cells. But I think more burning was the fear that we are now playing God and we are cloning people, and that things are coming up that are not really controllable. And this is why I think the society those days was really engaged, and only a couple of years, only 4 years after the first description already, the Bundestag has then passed the embryo protection law that said that it is only possible to use human embryonic stem cell lines in Germany that were generated outside of Germany, because until then there were no lines, and they were generated before this cutoff date of the 1st January of 2002. The idea is that no further embryos would be killed to generate human embryonic stem cell lines. And of course, there was additional regulatory constraints that you need to exactly describe what this is used for. There was an ethics board that agreed on all the research. And for commercial entities, it was not permitted to perform research with human embryonic stem cells at all. So this worked okay. I think it was a compromise that at least it was feasible. It was few groups in Germany that went this way and then applied for working with human embryonic stem cells lines. However, the researchers quickly found out that the few embryonic stem cell lines generated in these first 4 years, and you can imagine these were relatively few lines, some of them may have mycoplasma contaminations or other contamination. Anyway, that researchers found that this is not really competitive anymore. There are standards internationally on other iPS embryonic stem cell lines, and therefore there was an amendment in 2008 that it should now also be possible to use embryonic stem cell lines that were generated before 1st of May 2007. Now, this is still how it is today. So, long story short, no commercial entity is allowed to work with human embryonic stem cells in Germany. And if you want to use these cells, you are only permitted to use established lines before 1st of May 2007. And you can only do this when you exactly describe what you want to do, and this needs to pass a specific ethics committee. So it's feasible, quite cumbersome still. So the question is, first of all, maybe, do we need human embryonic stem cell research at all? Because as you know, we have now also the possibility of reprogramming any cell virtually into a type of similar to human embryonic stem cells, and the so-called induced pluripotent stem cells. By overexpressing specific transcription factors, we can really convert cells back into a very similar state to human embryonic stem cells. So the first question was, do we need this at all then? Or should we just switch to iPS cells at all? And I think that the students also agreed and also share this opinion that as as a reference, embryonic stem cells are really important because this is what really happens in nature, and this is the benchmark that we are trying to reach with our in vitro reprogramming research. Therefore, I think it is really good to have this option to work with human embryonic stem cells. And that said, also in other countries that where it's much easier, there are even clinical trials with human embryonic stem cells. So the second question is this law still a good compromise? And here I think the audience was mostly the idea that this is completely outdated for several reasons. First of all, we can only use these very old lines nowadays. It also was anticipated that, the company research is not necessarily bad research, so it should also be permitted. It's also helpful for society. And last but not least, there was concerns that, of course, every country anyway has its own regulations, so maybe this should be harmonized. Another comment, I think, on this question is that in Germany at the moment, we are relatively not research friendly, let's put it that way. So for example, in in green research of gene technology, or also if you look for animal studies. So I think there's this tendency to over-regulate things and rather not permit research openly. And therefore, there were also concerns that if we touch this embryo protection law again, there may also be now new concerns coming up. At the moment, many aspects are not regulated at all, which obviously is also not a good idea. But the fear was that if we open this box of Pandora, it might become even more difficult to do research in this direction. The next topic we talked about is then cloning. The first cloning was even done a little earlier, but the sheep Dolly was first cloned by Ian Wilmut. So this is then, let's say, an identical sheep born. Cloning is now, you can clone your dog and your cat by companies like Copycat or CopyDog, and also apes have been cloned. So I think the first question is how do you feel about cloning of animals, and then about humans? And for animals, I think most of the students said, yeah, but this is not helpful. Why should we do this? And then we discussed that there may be, some people who really lost their animal, their dog, and they loved their dog and they want to have it back again. And then under these circumstances, they felt, why not? Can be done. It's not really harmful, maybe, as long as the dog doesn't suffer then, which to today's knowledge, I think is okay. So this was commonly accepted. For cloning of humans, obviously, there was more discrepancy. Most of the students were strictly against cloning humans. That said, there have been attempts a couple of years ago in Korea that claimed that they had cloned, but I think so far there's no human cloned. The benefit, of course, is also not so really clear, because so usually the clone would not become better than their parents. Anyway, and there were also concerns about if we clone, then maybe it would be possible to use the organs then for the other donor. There were scenarios that we discussed where it may be helpful. For example, if I have a severe leukemia, there is no donor that could provide an allogeneic hematopoietic stem cell donation for me, and then under this scenario, it might be feasible that if I clone myself and I wanted to have a child anyway with my wife, then I could use the cord blood of my child, which would have this exactly same genetic background for my hematopoietic stem cell transplantation without harming the child. And as I said, we wanted to have the child anyway with me and my wife. That was a scenario where some people then said, yeah, okay, in this circumstance perhaps would not be so bad, but I think in general the audience was, of course, strictly against cloning of humans. The next point we talked about is about regulatory requirements now and then. And as an example, I just wanted to show here the development of stem cell research at all. The concept of hematopoietic stem cell was already, let's say, discussed by the end of 18th century. It became evident, really, or proven, that hematopoietic stem cells exist by this landmark experiment by James Till and Ernest McCulloch in 1961. And what these researchers did is they took mice, isolated hematopoietic stem cells from the bone marrow, and transplanted these hematopoietic stem cells, or this bone marrow, into other mice that were lethally irradiated. Now, the irradiation results in insufficient blood formation, and with the transplantation, these mice survive. And you can really then see that the engraftment can recapitulate the whole hematopoietic system. And even more interestingly, the Till and McCulloch then showed that it's possible to do serial transplantations from one mouse into the other. So after transplanting into one irradiated mice, they took the bone marrow and transplanted it to the next irradiated mice, and again, and they could thereby demonstrate that there is a subset of cells that are really capable to have enormous self-renewal to replenish the stem cell pool, and at the same time giving rise to all of the hematopoietic stem cells. And these experiments then were further fine-tuned, and you can even do later single-cell transplantation experiments. So this really proved the existence of functional hematopoietic stem cells. Now the question was, if this was described 1961, when do you think were the first human hematopoietic stem cell transplantations done? And the answer is actually pretty much at the same time. Already in 1958, even before this landmark paper came up, Georges Mathé in Paris tried to do the first allogeneic transplantations in let's say people that were treated for irradiation accidents. I think none of them really survived, but they, of course, already learned the first bits on how this may work. And Donald Thomas, who later also received the Nobel Prize, then I think was capable of really doing the first allogeneic bone marrow transplantation that survived in twin siblings. So this indicates that those days, research and clinical application were very close. It was virtually done at the same time. And the question was whether this would have been feasible with today's regulatory requirement, because as the students have learned in this course, there are many regulatory hurdles nowadays. You need to really have exact clean rooms, precise protocols. You need to have a whole list of safety requirements and proven therapeutic, let's say, concepts that you feel that if I do this treatment in a trial, this is as safe and as reproducible as possible for the patient. While this is good on one side, the requirements are so high that it is virtually impossible now for a university hospital to perform such clinical research, because it literally takes billions of euro to have such a study ongoing, particularly if it's really having a cellular product where you really need to keep cell and culture under GMP conditions. So this is really expensive and really a long process. This can easily, of course, take 10, 20, 30 years. So the question really is, where would we be if those days the regulatory requirements weren't there? And on the other hand, are we harming people by not being able to develop new regulations so easily at this point? And there the audience was also quite, let's say, diverse. Many people said, no, but these requirements are really good and we need them to really have it as good and as reliable as possible. But there were also concerns that indicated that maybe for phase 1, 2, and maybe even phase 3 trials, the hurdle should be much lower to be able to generate new treatments and to first see what's really working to bring development faster on a timeline. So I think this is something that society also needs to deal with. It is always difficult to reduce regulation, but perhaps society would benefit if we would, particularly in the beginning, make some of these approaches a bit easier. The next topic we talked about is somatic gene therapy. This was also taught in this lecture series. It's actually quite remarkable that it's now possible to use the CRISPR-Cas technology to repair mutations or gene defects. For example, for beta thalassemia or sickle cell disease, it's possible to modify hematopoietic stem cells in a way that they express an embryonic hemoglobin gene again, and then the disease is more or less cured. So it's possible for these patients to have their stem cells isolated. They are then CRISPR-Cas modified, transplanted back in the patient, and this is still a relatively new therapy, but so far it really looks outstanding. The patients don't need to have transfusions anymore, and this really looks nice. So while there generally are not so many concerns about this somatic gene therapy, there were some critical concerns about the costs, because the costs are really quite high. For a single treatment of one patient at the moment, the FDA is judging about 2 to 3 million euro per treatment, which, if you sum this up, of course, is a high financial burden for the society. So I think it's also something that needs to be discussed in the future with these upcoming technologies, also with CAR T-cell technology that I think also has fantastic therapeutic results. And for many malignant diseases, society needs to discuss how they are coping with these high costs. We are then discussing that maybe the costs are going down if really this is off-the-shelf product, and maybe CAR T-cell therapy in the future is really used for thousands of people at the same time. Then production costs can also go down significantly. But still, I think this treatment will never be, let's say, cheap. There will always be quite expensive, and therefore talking about the costs and what is normally covered by society is something that sooner or later I think needs to be done. So anyway, the next critical point is human germline modifications. So this is different to the somatic gene editing that I just mentioned before. In this case, we are really modifying the germline cells. So, for example, sperm or egg or the early embryo, and then this is a modification that is not only changing the inhibidium, but can also change their offspring and their offspring again. So we are here really changing the human genome that is inherited later to others. And of course, we are able now to modify not only specific cells, but we are really changing all cells of the human at the same time. And while this may sound like science fiction, it was already done in 1918 by a scientist, He Yankui, in China, who modified two girls to disrupt the CCR5 receptor. And the idea is that this receptor is important for HIV infections. In fact, there have also been somatic gene editing trials before, and a lot of research that maybe if this receptor is not so super important, and then so there were also already ideas that perhaps if this receptor wouldn't be there, there's less HIV infection. In fact, there's also some normal people that have mutations and they are also less susceptible to HIV. So the rational principle is there. But you can imagine that there was a huge outcry, and when the scientists came just a month later to the Human Genome Editing Summit in Hong Kong, there was a huge arousal, and there were really not standing ovations for this. This may also be up because how this whole thing was done. It was just not, there was apparently not, this was not openly discussed. There was no ethics committee largely involved, and the study was ultimately published on YouTube, with a video where the scientists said, "Hey guys, I made it." So of course this is a very fast way to have your preprint out, but it's also not very scientific. And all in all, I think there was a huge outcry, and that scientist also lost his job then. And to my knowledge, since then, human gene line modification has not been made public again. There was also many, of course, countries also within the World Health Organization that discussed how to deal with human germline modifications in the future. The difficulty is, of course, that you may correct some diseases. In this case here, I think there's common sense that HIV that is, first of all, it's treatable, and secondly, this is only the perception to receive the disease that you can anyway protect yourself of. So this was not considered as, let's say, good enough reason for this germline modification, particularly also since there are studies that indicate that the receptor ultimately may also be beneficial for other diseases. So in the end, I think the benefit here is not really good enough. But if there was a really good clinical reason, then there may be circumstances circumstances where perhaps this in the future may be possible. Another difficulty is that there may also be enhancement in the future. Perhaps in close future we have the possibility to modify the germline in a way that the offspring will become more intelligent, or they are protected against Parkinson's disease or cancer. At the moment, this is scientifically not feasible to my knowledge, but it's not completely off the track. I think this could come relatively fast. And then the question, of course, is whether or not this should be permitted, because the offspring might be really enhanced. While the general concerns there was that we really shouldn't do this, we should not play God and not modify our germline, there was also a consensus that if we do not allow this in Germany, it may be allowed in other countries. And if that's the case, there may also be an imbalance if suddenly other countries have the more clever or the better protected offspring. There may also be two reasons to these countries, particularly, of course, within Europe, where the borders are so close. So we are only 5 kilometers to the Netherlands and to Belgium. So there are certainly difficulties also in how this is governed by regulation. Anyway, the German Ethics Council has also dealt with this in a, I think, quite nice paper that they published in 2019. I'd like to highly recommend this because it gives a very nice scientific background and also some ethical considerations that I think is really worth reading. And the general perception is that, surprisingly, although also on this board were some people from the churches, there was agreement that this is not categorically, under all circumstances, not allowed in the future to have human germline ethic in Germany. But of course, at this point there was very little need seen, and the ethical standards need to be really high to bring this over. So for example, there should be precise ethical standards that are taking care of this. Ideally, also with an own review board and globally scientific ethical board that takes care of this. There should also be global rules that apply for this. Of course, this is not getting easier with the, let's say, political development over the last years. And there needs to be a really good need. So enhancement was also not considered to be a good reason for human germline ethic by this council. But there may be, and this is also, let's say, more a theoretical construct, there may be clinical needs where this may be useful in the future, but then we need to have it really efficient and safe. And that's also a downside described 2019. I think we now already have a little more insight into the CRISPR-Cas technology, but you always have some off-target effects that may also result in other mutations, of course, genome-wide. So we need to make these tools more safe and more efficient and have better safety nets to really make sure that there is no harm. And last but not least, there was also the request for an international moratorium that at the moment these germline interventions shouldn't be done in human anywhere around the world, which I think is shared by most countries. But there was a big arousal lately that maybe South Africa changed their regulation a little bit and that they didn't permit it now explicitly anymore. So while I think that may also be a little, well, let's say there was no clear evidence that would start anything like this, I believe that countries in the near future might go this track, and I think the society needs to reconsider then how they deal with this and how also maybe legislation needs to take these issues into consideration. Another point is then that it's possible to have chimera. Of course, we can have chimera are a mixture of cells from two different species. And of course, you can have experimental settings where you, for example, transplant cells from humans into mice. So this has been done, and we also learned some aspects here on stem cell biology. Of course, this is also something that ethically and also from animal welfare perspective needs to be heavily discussed. But these things are more or less there, and I think also important for clinical research. That said, there is also another line of research where we are now making chimeras by putting pluripotent stem cells together. For example, making chimera of embryonic stem cells from human and mice that, of course, are not viable at this stage. This has also been done with human and ape embryos. And there is research to learn how we can stabilize these embryos, that at least the first parts of embryo development can be done. So far, of course, these chimera have never really been brought into an animal. But the question really is this okay? And how should we allow this research? Is this dangerous for the future? And this question didn't cause so much arousal, because I think also for the students it was a little cryptic of why this should be useful and why this makes sense. It became a little, let's say, more vivid when we talked about something that is really now in the news and closer to a clinical application. And to make this clear, I want to briefly introduce let's say the scientific background. For example, the gene PDX is a master regulated gene for pancreas development. If you have a knockout mice for PDX1, this mice doesn't have a pancreas. Apart from that, the other organs are more or less there, so the mice can survive, but it doesn't have a pancreas. Now, if you inject into this early mouse embryo of a PDX knockout mouse other embryonic stem cells of a wild-type mouse, the mouse will generate a pancreas again, because those embryonic stem cells that were initially implanted into the embryo are filling the niche and are actually building the new pancreas in the mouse, which of course is quite, let's say, it's amazing how this works at all, right? How these cells are self-organizing to then fill this niche and reorganizing this organ. It is even more amazing that it's possible to take a knockout mouse and transplant embryonic stem cells from a rat into this. And then the pancreas will be derived from rat cells entirely, which I think is of course, not only that this is an interspecies chimera now, it's also, I think, again, remarkable how well then the communication and differentiation of these cells is protected, that this is still feasible. What is even more remarkable is that you can then take this rat pancreas from your knockout mouse and transplant it back into another rat. And this is actually the scenario that is now more or less in the news, because as you know, we have a shortage of many organ donors, maybe also because of the regulatory hurdles that we have in Germany that prevent us of having more donors for organs. Anyway, so the possibility might be that we are creating knockout pigs from PDX1, and then we are generating these let's say pigs that usually wouldn't have a pancreas, but then we are complementing with, for example, induced pluripotent stem cells that were generated from patients. And then this pig would develop a human pancreas, and this pancreas could be transplanted back into the patient to replenish the pancreas function, including also, for example, treatment of diabetes. From animal experiments, we know that this in principle is feasible. These mice also exist, these pigs also exist. So in principle, such research is possible. And it's not only for pancreas, we can do similar things also for many other organs. And while this is not so much driven here, for example, in Japan, this is pursued, of course, also with a lot of ethical debate and with interaction with the society. But this is something that I believe may come also swap back also to Germany, and that also our society needs to deal with this question, whether or not they want to have chimeras to generate human organs in the end. The audience was quite mixed here. So there was some said that, yeah, okay, but we eat pigs all the time, and this is not, the pig is not really harmed, so there were not so high concerns about this. Other people said, yeah, but this is not good, it's not normal, and also maybe we don't know whether we humanize the pig in the end. Maybe there are some neurons or even some germline cells that would then, human germline cells that would then develop in the pig, which I think is quite unlikely that this is feasible anyway, but these are of course concerns that need to also be taken into consideration, and this really points that these are questions we need to address. Next question goes towards synthetic embryos. And here I just want to briefly show the normal embryo development. And that's quite remarkable. Actually, you can see here on the upper scale the mouse and lower than the human embryo development. And this comes with a little different time scale, but you can see that after day 7, 8 for mice and after 14 days for humans, we already have quite complex human structures. And now from a stem cell perspective, it's really fantastic to see how these pluripotent stem cells, whether they're embryonic stem cells or induced pluripotent stem cells, are capable to self-organize into organoids and into embryo-like structures. So the easiest structure is a cell aggregate. If you just take these cells together, they form an aggregate that is in solution, and we can then see that in this aggregate, upon maybe differentiation also after growth of the aggregate, there are certain regions that are more going towards endo-meso-ectoderm, and that there is already in this very, very, let's say, aggregates, not really an embryo, of course, there are sulfate decisions in certain regions that indicate there is some self-organization. You can enhance this if you use specific Wnt inhibitors or other chemical compounds into so-called gastroloids, or it's also possible to even go a step further by mixing not only the very primitive embryonic stem cells, but maybe also a little more differentiated cells towards the trough ectoderm, that you have more complex structures. And now recently it became possible to have what in the literature is called synthetic embryo. And this came up in the year 2022, so it's not so super long ago in mice, and a year later it was also described for humans. If you pre-differentiate these cells towards trough ectoderm, primitive endoderm and embryonic stem cells, and you mix them under certain culture conditions, you can really generate these kind of synthetic embryos that recapitulate much of the anatomic morphology we have in normal embryogenesis. This is depicted here on the right side, where you can see on the lower panels the normal embryo developing. You can see on the left the neural fold, the neural tube, and then some somites indicated by the arrows. So this is how a normal mouse embryo would look. And on the upper you can see how this in vitro generated aggregate of cells of these different cell types under appropriate differentiation conditions would also differentiate. And they also form a very similar structure with a neural fold tube and somites. So you can see that really these cells have an enormous potential to self-organize. And that, of course, while it's fantastic to see from a stem cell perspective, I think there's a fair question of, should we allow such research, and to what stage, and what are then the rights of a synthetic embryo, and when does the synthetic embryo receive these rights? So of course, at this stage, maybe it is still just a clump of cells, but it's a fair question that society needs to deal with in the future. What we really need to learn from such research, maybe of course this can be important to generate better ways of enabling people to get children that wouldn't be able with normal ways. But on the other hand, of course, this is very critical with regard to many aspects of our society. So I think this is currently heavily discussed, how this should be controlled, and how maybe there are thresholds that shouldn't be crossed. So these were the questions that I touched in this lecture on cell and gene therapy. As I mentioned, this was a very lively discussion. I want to thank the students here for contributing. It was really, we were talking for 3 hours quite excessively about all of these issues. And I hope you also enjoyed this. Thank you.